This was an exploratory study looking at novel biomarkers to try to define if they would be useful in different stages of heart failure. So principally we took patients with known heart failure, either symptomatic or at risk for heart failure, and we obtained serum samples, plasma samples that we then looked at for different biomarker levels.
Kidney dysfunction is very common in patients with heart failure. In fact, about 20 to 60% of patients with heart failure have chronic kidney disease and is associated with worsening morbidity and mortality in these patients. So there's a lot of interest in measuring biomarkers that may be able to detect kidney dysfunction earlier than some of our current assays. So our traditional biomarkers that we use, such as BUN and creatinine, they are not very specific or very sensitive. And in fact, by the time they become elevated, the disease process has already progressed to a significant degree. Therefore, novel biomarkers such as kidney injury molecule 1 or endothelin 1 may be able to detect patients who have renal dysfunction at a much earlier stage, and that could identify patients who would potentially benefit from treatment earlier. The current measurements of KIM1 has been done in the urine, but there are a lot of trouble with using that kind of an assay. Urine is not very commonly used for typical tests, and it's not as stable, and it's not able to be measured as precisely. The Singulex plasma assay is a very precise test that is able to detect biomarkers that are very present at very low concentrations. Our usual assays in the past were able to measure concentrations of biomarkers that were present in patients who had higher levels, such as those who have had disease that has already progressed. With the Singulex assay, we can measure much lower levels of biomarkers that perhaps are present in patients who have subclinical disease. Thus, we can use the Singulex assay to be able to detect patients at a much earlier stage, and those patients would potentially benefit from earlier treatment. Traditionally, renal function is assessed by changes in glomerular filtration rate, and we break patients down into certain stages of GFR based on the degree of renal dysfunction they have. And we were able to find that patients who have progressively worsening renal function have higher levels of plasma KIM1 and plasma endothelin 1. KIM1 has to date not been measured in plasma, and our assay showed that KIM1 is significantly correlated with a change in renal dysfunction with a very low p-value. And we think that KIM1 is likely to be a very strong predictor of patients who have heart failure who are going to have worsening renal function over time.